Hi again, everyone, and welcome back to our first session at World CRISPR Day on cell and gene therapies. So here to chair this exciting session featuring scientists from across Europe and the United States is another CRISPR pioneering scientist, Dr. T.J. Cradick, who is currently Chief Scientific Officer at Excision Biotherapeutics. TJ has made numerous contributions to the genome editing field over the past 16 years, both through his pioneering work with targeted nucleases in academia at the University of Iowa and Georgia Tech, and in industry at both Sangamo Therapeutics and CRISPR Therapeutics, where he acted as head of genome engineering and was instrumental in launching one of the world's first CRISPR gene editing clinical trials for sickle cell anemia and beta thalassemia. TJ, it's great to have you here. So please join me in welcoming Dr. TJ Cradick, and I'll hand it over to you to chair the session.
Wonderful. Thank you, Kevin. Excited to be here and hosting this cell and gene therapy session. As we're running a little late, I'll keep it short and just mention that the Q&A tab should be available, and it was recommended as the way to put some questions in, and we'll get them to the speakers and they can address them. With that, I'll jump to introducing our first speaker, Tanja Rothgangl, a Ph.D. candidate in Gerald Schwenk's lab at the University of Zurich, where she focuses on the improvement of gene editing tools and their applicability to in vivo gene editing, including use in adenine-based editing to permanently lower LDL-C levels in blood of mice and non-human primates. The study was recently published in Nature Biotech, and with that, I'll introduce her and allow her to go forward. Thank you.
Thank you very much for the introduction. Okay, good. So I'm very, very happy to start this session, and I will talk about in vivo adenine-based editing of PCSK9 and macaques, which reduces LDL cholesterol levels. And so I actually would like to split this whole topic, this whole project into three questions. One question is, can we permanently cure monogenic diseases? And so starting with this, I would like to quickly tell you what I understand from monogenic diseases. So they're caused by just one gene. And in our project, we actually are interested in the diseases that are just caused by a single base. And what happens is when you have in the whole DNA, like one wrong base in the whole genetic code, what can happen is then you have a dysfunctional protein. So you have the wrongly folded protein or different levels of the protein. And the solution to the whole problem would just correct the code, right? So you would just correct the base. And in our case, the problem would be familial hypercholesterolemia, which leads to elevated low-density lipoprotein levels. And the solution would be adenine-based editing. Just as a quick introduction, I think here in the audience, I do not have to introduce you to Cas9 nuclease, but as you all know, there's two ways of the repair of the double-strand break of the DNA. So non-homologous end joining, or also homology-directed repair. And with homology-directed repair, you would be able to say, yes, you can cure such a single base pair change. But unfortunately, this really requires dividing cells. So there was from the Leo lab, there was now really a great invention, base editors. And what they did is they used the Cas9 ligase and fused the deaminase to it. And then what happens, what you can do is, so this deaminase that deaminates the adenine to inosine. And then the inosine is red. So at the opposite strand, there's a nick, and the nick gets resected. And then this nick or the DNA is repaired using the inosine as a template. And this is read as a guanine. So you end up with a GC base pair when you started with an AG base pair. And now this big advantage here is that it works in non-dividing cells. So using this tool, our first challenge was, can we bring this thing in vivo and does it work in vivo? And we tested two different delivery methods, adeno-associated virus and lipid nanoparticles, LNPs. And for starting with adeno-associated virus, we actually tested different adenine base editors. And what we saw is that we can get very efficient editing in the liver. And actually with AB max, we get very high editing rates, 60% in the whole liver, which equals like above 80% in the primary hepatocytes. But the indel rates were lower in comparison to, for example, the ABEAD. And this actually then also results in a significant reduction of the PCSK9 levels, which also result in the reduction of the LDL levels. And then looking at the LNPs, we're actually able to really like scale this up in a dose-dependent manner. So the more you inject, the more editing you get. And also the PCSK9 levels and the LDL levels can be reduced using LNPs. What's also important here is that we can sustain this editing effect with both delivery methods for several weeks. So, okay, so it seems like, yeah, it's possible, right? You can most likely permanently cure monogenic diseases, but how safe is this approach? So when you compare AAVs with LNPs, with the AAVs, we saw that we actually have the expression. So here we co-express RFP with the base editor, and we have the expression over several weeks. We basically don't lose the base editor. The base editor keeps being expressed over several weeks. When you look at the LNPs, the story is very different. You basically have just a period of time where the base editor is expressed, and then after 72 hours, we cannot detect it anymore. However, the editing is there. So you just need a very short period of time for the edit to occur, and then the base editor is gone again. And additionally, what we saw is when you look at the adaptive immune response, we actually saw antibodies to the base editor when we used AAVs, but we did not see any antibody response when you used LNPs in mice. Then we further looked into safety. So there was this concern that base editors might induce RNA off targets. So they would randomly deaminate adenines on RNAs. And we actually saw that in vitro as well. So if you look at the first two bars or areas here, you see the in vitro analysis. And we saw that in vitro, we actually see a lot of unspecific adenine edits that were not intended. But when you look in vivo, we actually see like far less, and only also with LNP, we just see it for a short period of time, and it's gone after a week for sure. And we explain this because the dose is far lower. So in vitro, we have like almost like 80% of the transcripts are actually our base editor. So obviously, you would expect off targets if you highly overexpress such a base editor. But if you look in vivo, our expression levels are far lower, and this probably also explains why we see less off targets. But nevertheless, you might say, okay, RNA-seq is very noisy. There's a lot of transcripts. We might not see everything. So the more important question is actually what happens on the DNA level. So what we did here is we took the primary hepatocytes from the mice, which were edited, and then we clonally expanded a single hepatocytes. We did this because if you think about bulk sequencing and there would be an off-target that is very rare, you would not find it in the noise of all the other DNA from other cells. And if you would do single-cell sequencing, you also have a high error rate by the method itself, so you would also not find it. So we thought we just exploit nature itself, and we take the cell, let it expand, make accurate copies of its DNA, and then do whole genome sequencing. And what you then get is a so-called 96 nucleotide graph from each individual clone, where you see the different conversion rates within the whole genome and the DNA that we find. And if you look at the fifth column here, you see the T to C conversions, which is also including the A to G conversions, which are the conversions that should be done or should be found when you use adenine base editor. And when we now compare this to control clones, AAV clones, and LNP clones, we actually did not see any increase of these edits. So we could not find any edits on a DNA level. And then to even further prove the safety, we came up with a long-time study in a sensitized mouse model. So we had mice that had a knockout of P53, so the major tumor suppressor gene, in the hepatocytes, and we confirmed this, that the P53 is really gone. We injected them with the base editor, so we made sure that it also worked, the base editor was in there by checking the PCSK9 levels. And we also checked the editing in the end. And they really had, for over a year, they had base editor in. And then we looked at the livers, and we even gave the samples to a pathologist. And we really did not find any signs of any cancer formation. So this long-term study even further seemed to show that there cannot be a lot of off-targets happening that would be very detrimental. Okay, so we think the approach might be pretty safe. So now the next question was, can we translate our findings? So would it be feasible for clinics? And for this, we actually were able, so I have to thank here already Acuitas Therapeutics, because without them, it would not have been possible to do this big non-human primate study. And here we tested with the LNPs whether or not we can have efficient editing in a large animal model. And what we saw is that we actually achieve it. So using 1.5 mg per kg, we actually have editing rates of around 30%. So these editing rates are high enough to significantly lower PCSK9 and also LDL levels. And then we further looked into the innate immune response and also liver toxicity. And what we saw is like right after injection. So if you look at the gray and the dark yellow bar, these animals got a second dose. And every time after injection, you got like a very quick increase, but it goes back to baseline very quickly. So this again speaks for the LNP approach, because it is so transient that you just need it for a short period of time, but then after editing it happened, you don't see any side effects anymore. We also here looked at adaptive immune response, and interestingly, actually here with the LNP approach in comparison to the mouse approach, we saw adaptive immune response to the base editor. So this underlines furthermore that a large animal experiment is important, because the immune response in larger animals is probably more sensitive in comparison to rodents. So yeah, so altogether, I think I could show you that it probably is possible to permanently cure monogenic diseases. I would think it's a pretty safe approach in comparison to other approaches out there yet. And I think it's also possible to scale it up, and theoretically it would be feasible for the clinics. And with this, I would like to already acknowledge the people that were helping. So I would like to thank Melissa, Paulo, Ying, and Sean for the help there from Acuitas Therapeutics and made this big study possible. And also would like to thank the people from Syntago that helped with the production and also the large and also the small scale experiments with providing the guide RNAs and different modifications. I would like to thank the people that did the bioinformatic analysis, and also the people from my lab. And first and foremost, don't want to forget to thank Gerard. He is my PhD supervisor, and with him, obviously, without him, it would not have been possible. And yes, I would also like to thank the funding and you for listening, and I would be happy to answer any questions.
Well, you received a whole pile of questions. I know you're probably too many to scroll through in the short amount of time. One of the questions was on utility for primary cells and such. I think you've got some believers that hope to jump on this.
Sorry, it's in the public. Can you repeat it? I think I don't see them.
Oh, one of the, okay. So I will look to try and transfer them over. But the question was, is it possible to use in primary cells? Was one of the early questions that came in.
Oh, yeah. Okay, yeah. So actually that's something that we had to overcome, because we could not expand primary hepatocytes. We actually had to use a protocol that was published by the first author is Katsuda, and it's a cell paper, and they show that you can convert them into liver progenitors. They call it chemical-induced liver progenitors. And you give them a cocktail, so you actually have an intermediate stage. It's not really stem cells, but it is like a progenitor cell from the hepatocytes. And in this stage, you can actually expand them. Yes.
And so the second question, which is probably all we have time for, is you show one set of data, and now they want to know what's the consistency between batches.
I don't know what we are actually talking about. I assume it's probably the whole genome sequencing and the RNA-seq data where we have different batches. So we always took different animals when we compared. So then the variations, for example, for the RNA-seq was very similar, even though it was different animals. So we found like similar amounts, even though it was different off-targets, we still saw a similar off-target amounts that we could detect.
I guess the question was in terms of the LNP and such. The question was how consistent was the final product?
Okay, I'm sorry for. Okay, thank you for this. clarification. Yes, no, that actually with the LNPs, so we tested also different guide RNAs. So we always had like the same mRNA of the base editor, but then we also complexed it with different guide RNAs, and we really got it very robust. So the animals were also not injected always at the same day, and we also did the same experiment sometimes at another day, and we could observe the same result. Absolutely, it was very, very reproducible working with the LNP.
Wonderful. Thank you so much. I
Yeah,
appreciate the presentation and the answers. I would like to jump now to our second speaker. Dr. Demetrius Wagner is head of the R&D team at the Berlin Center for Advanced Therapies, BCAT, at Goethe University Hospital in Berlin. BCAT is an academic institution committed to translating innovative research by GMP into early clinical phase 1 and 2. He's also a junior lab member at the Berlin Institute of Health, where his group is focused on establishing and testing gene editing platform technology for cell therapy. With that, let me introduce Dr. Wagner.
Thank you very much, TJ. And thank you very much, I would like to thank the organizers a lot for giving us the opportunity to present a small overview on the clinical pipeline that we have at Berlin Center of Advanced Therapies, where we use gene editing to engineer designer T-cell immunotherapies for transplant medicine and oncology. So these are my potential conflicts of interest. So in the center of many of the projects that we do at BCAT is adoptive T-cell transfer. And the general idea here is that we can use the patient's own immune cells to generate a therapy. And we can do this by drawing blood and isolating specific T-cells that, for example, are specific for a virus or have other functional properties that we want, and then activate and expand them outside the human body, use genetic engineering to then formulate a very potent cell therapy product, which we can infuse back to the patient to either resolve an immune-mediated disease or, for example, treat a disease like cancer. So I started working with CRISPR back in 2015 during my time at Baylor College of Medicine, and there we followed the work of Alex Marson and found that if you co-electroporate CRISPR-Cas9 ribonucleoproteins, short RNPs, so recombinant Cas9 and single guide RNA into activated T cells, that is a very powerful means to genetically modify T cells. And I'm going to give you two examples on how we can use this clinically. One, first step, it's easier, knocking out a gene in T cells. We can use this to engineer drug-resistant T cells. And then later on, I'm going to give you an example how we can use knock-in, so inserting new stretches of DNA in T cells. So let's jump right in. So my boss is Petra Reinke. She's the director of BCAT and also a transplant nephrologist. So her whole life, she is dedicated to treating patients with end-stage renal disease that require a kidney transplant, either from a healthy living matched donor or a deceased donor. And her primary focus in her work was to avoid that her patients reject the kidney graft. And this rejection is usually due to an alloreactive immune response by T and B cells toward the foreign tissue of the kidney. So we use very potent immunosuppressive drugs to prevent this. However, from the immunosuppressive drugs arise other challenges, and these are the side effects or damage that is associated with immunosuppression. And the most important complication of the immunosuppression are severe infections, for example, by reactivation of latent viruses such as cytomegalovirus. And over the last 2 decades, adoptive T-cell transfer with virus-specific T-cells has become a very important modality to prevent and treat severe infections like this. So what we want with this virus-specific T-cell is that after infusion, it expands and persists in the patient to fight the disease and makes it or transform a patient into a healthy individual. However, the systemic immunosuppression will also affect our T-cell product. We already know that the immunosuppression that patients with solid organ transplantations receive reduces the T-cell persistence, and we have indications that in patients with extensive disease, they prevent effective antiviral control. So triple immunosuppression is the gold standard, and there are three pillars: the steroids, nucleotide synthesis inhibitors, and calcineurin inhibitors. And we will focus on the last. Calcineurin inhibitors are small molecules that act in complex with so-called cyclophilin proteins, and the complex of the calcineurin inhibitor and the cyclophilin then inhibits the calcineurin phosphatase and prevents translocalization of the nuclear factor of activated T cells. This reduces the amount of effector cytokines that these T cells can release and limits the availability of other growth factors. So cyclophilin proteins are well studied. One cyclophilin protein is just called cyclophilin. This is the target of the CNI cyclosporin A. And the second is FK506 binding protein 12, or short FKBP12. And that is the target of tacrolimus. As tacrolimus is our favorite calcineurin inhibitor, we wanted to know whether we can edit the FKBP12 gene using gene editing to make the T cells resistant. So this work was started when I was still in medical school working in the lab of Michael Schmück-Henneres, and together with Laila Amini, who did the lion's share of this work, we established a GMP-compatible process for applying gene editing for virus-specific T cell product. So for this, we isolate peripheral blood cells and then stimulate the cells with viral peptides and then select the T cells that start producing interferon gamma, a key effector cytokine. This allows us to enrich for virus-specific T cells and reduce the amount of unspecific but also alloreactive T cells in our T cell product. Then 7 days after expanding those cells, we can electroporate them with hi-fi Cas9 and synthetic FKBP12 specific guide RNA. And within our established process, we have efficiency of over 70% on average. When you take those edited cells and compare them after stimulation with unmodified controls, you can see that if you stimulate with ZMV peptides, they produce large amounts of TNF-alpha and interferon gamma, which is indicated by this cloud of cells in this upper right quadrant. You can see that's very similar in the edited cells on the lower row and the upper row, the unmodified controls. However, if we do the same stimulation in the presence of tacrolimus, you can see that the unmodified control are unable to produce cytokines, while the edited cells are still potently expressing antiviral effector cytokines. Similar effect is also seen when we have the cells in triple immunosuppression, which indicates that these FKBP12 edited cells will have a therapeutic advantage. Importantly, if we do the stimulation in the other calcineurin inhibitor, in the presence of the other calcineurin inhibitor, cyclosporin A, both of the cell types are inhibited. And that is great because it is an inbuilt safety switch. So if something is going wrong, we can switch the patients from tacrolimus to cyclosporin A, and cyclosporin A is a clinically approved drug with similar efficacy in preventing graft rejection. So the tacrolimus-resistant T cell platform has dramatically moved from preclinical development into the GMP, and we're currently doing our final validation of our GMP process with GMP-grade materials and hope to start a first-in-human clinical trial very soon. So let's go to the next step. For me, the logical follow-up after a knockout is, of course, whether we can also use this technology to insert new stretches of DNA. And here we wanted to follow again Alex Marson's group, who has shown that if you co-electroporate RNPs and synthetic DNA donors, you can actually efficiently knock in new stretches of DNA. And we wanted to use this technology to redirect T cells with tumor-specific receptors for patients with cancer. Jonas Kart, an MD and a PhD student in my lab, took on this challenge. Over a year ago, he started working on generating a virus-free platform to create these TCR-replaced CAR T cells. For this, he designed an HDA donor template that allows us to insert a new tumor-specific chimeric antigen receptor in frame with the TCR alpha chain locus. So after successful editing, what we would expect is that we have a dysfunctional truncated TCR alpha protein, but instead we have a fully functional CD19 specific CAR, and the CD19 specific CAR has a CD19 binder, and after engagement with tumor cells, this transfers co-stimulation and a TCR-like signal to the T cells. And in total, this means we can redefine the specificity of the T cells towards CD19, and in the same way, we eliminate potentially alloreactive T cell receptors from the surface, which would make this technology amenable with, for example, an allogenic off-the-shelf products where we generate CAR T cells from healthy human donors. So Jonas has perfected this method, and it works like this. He isolates T cells from patients, stimulates them for 2 days, and then performs the electroporation of the RNP with a double-stranded DNA donor. And then as early as 2 days, you can check the cells via flow cytometry. Of course, unedited T cells, they always have the T cell receptor, but they don't have the CD19 CAR. If you electroporate guide RNA Cas9 and the double-stranded DNA donor, you can get around 20% of the cells have the CAR but lack the TCR. So here the knock-in was successful. We were not happy with these results and tried to optimize it and collaborated with IDT, which provided us with a small molecule compound that increases homology-directed repair. And with this RHDR enhancer version 2, we can almost double the knock-in efficiencies in these cells. And to even further increase the efficacy, we identified a cocktail of two DNA sensor inhibitors, which we can pre-treat the cells with, and the combination of HDR enhancers and DNA sensor inhibition allows us to knock in CARs at rates above 50% on average. So this is a great technology to generate CAR T cells, and it is also nice that they're potentially amenable with an off-the-shelf approach. But since there are already a lot of CAR T cell products out there, we have to compare it with the gold standard. So we wanted to assess the performance of our gene-edited CAR T cells in comparison with conventional CAR T cells that are produced by lentiviral transduction. And here we are very thankful for our collaborators in the Strippeck lab in Hanover, which did this experiment with our cells. They performed a xenograft mouse in vivo experiment where they took immunodeficient mice and infused luciferase-labeled leukemia cells, and then injected our CD19 specific CAR T cells 4 days later. Then over a stretch of 5 weeks, they continually measured the leukemia burden using bioluminescence imaging. So if you look in the first column, you can see that the mice that were injected with the tumor only, there's a dramatic increase in the tumor burden, and we had to stop the experiment at week 5. In the lentivirally generated CD19 CAR T cells, you can see that in the earlier weeks, they are able to control the leukemia, but at week 5, you can see some mice that suffer from extensive leukemia. The TRAC knock-in CAR T cells appear to have better leukemia control. However, at last time point, there's also an increase. And the data is summarized here on the right in a log scale. You can see that on week 5, there is no statistical significant difference between the tumor burden in the tumor-only condition and the condition with lentivirally transduced CARs. However, the gene-edited CAR T cells are still able or still have significantly lower tumor burden. And with this, I'm pretty much sure that I'm already out of time. As a last quick comment, I think it's amazing what we're currently seeing in the CRISPR field. Both knockout and knock-in projects are widely investigated, and we have multiple clinical trials already ongoing, and I'm excited for the future to see how we can combine this to make better T-cell therapies. And with this, I would like to thank you for your attention and have up here the name of all the amazing people making clinical T-cell therapies extremely hard. You need big teams and a lot of different expertises, and I'm very glad that we have this in Berlin. I would, of course, like to thank my growing team and the R&D team, but also at the BIH lab. And now I'm very happy to take any questions that you might have.
It does look like you have quite a number in the Q&A, but I'll summarize because several people asked overlapping questions about testing various forms of donor DNA. So maybe you could address several of the questions in the little bit of time we have, and talk about any experiments you've done comparing different single-strand, double-strand, AAV, et cetera, as your donor.
So when we started working on this, there were not many people who could make single-stranded DNA, which would be the useful alternative, at GMP grade. That's why we focused on double-stranded DNA and have not yet compared the two platforms. We are looking into that because more and more DNA providers are now looking into GMP production of single-stranded DNA, but so far we have not compared it systematically.
Wonderful. Well, I thank you. I wish we had more time. There will be questions available in the archive, although I'm still trying to look up how you might be able to reply to those, but we'll continue to work on that. Next, I'd like to present two people that are co-presenting the next session. Giorgia Santilli is the Associate Professor in Gene Therapy at UCL, Great Ormond Street Institute of Child Health, London, where she has worked for several years on the gene therapy for primary immunodeficiency. Recently, her work on lentiviral gene therapy into hematopoietic stem cells has been successfully translated into a therapy for patients with chronic granulosa disease. Co-presenting, Alessia Cavazza is Assistant Professor at UCL, Great Ormond Street Institute on Child Health, where she leads a research team focused on the development of gene editing platforms applied to hematopoietic stem cells to tackle rare genetic pediatric disorders affecting the immune system. So please welcome these two who will be tag teaming on the next talk. And again, put the questions in the Q&A. And at this point, it might be clearer if you address for whom your questions are, and that they might be able to follow up later. Thank you.
Yes, hi everyone. So as Thomas said, our lab has a long-standing interest in the treatment of primary immunodeficiency disorders. Those are intrinsic genetic disorders of the immune function that lead to absence of one or more blood cell types, or to the presence of blood cell types that do not function. So children are susceptible to frequent infections, and this can lead to death in infancy in the most severe forms. The diseases have similar symptoms, but different underlying causes, and over 300 causative genes have been identified. Just to remind you that all blood cells derive from hematopoietic stem cells. And so no matter which genetic defect you have, can be cured by replacing the hematopoietic stem cells with an healthy one, and this is what is called hematopoietic stem cell transplantation, or nowadays by actually correcting the patient's own hematopoietic stem cell ex vivo. So this is ex vivo gene therapy, where we take hematopoietic stem cells from the bone marrow or the mobilized blood of a patient, and then we culture those cells in the laboratory, where we add a viral vector usually to insert the missing gene into the cells, and then the cells are given back to the patients, where they home again in the bone marrow and give rise to healthy blood cells for a lifetime. So far, this gene addition has used retroviral vectors, and nowadays we are mainly using lentiviral vectors. And there are a few trials showing very exciting results for many PIDs. However, especially early trials have also highlighted the pitfall of this technique. And more importantly, the fact that those retroviral vectors have a semi-random nature of integration, so they are prone to insertional mutagenesis. And also it's quite difficult to reach a physiological level of the expression of the gene. So gene editing nowadays can actually counteract those problems, and basically we can use an endonucleases, and nowadays we're using quite a lot the CRISPR-Cas system to create a double-strand break in a chosen locus, and then harness the homologous recombination machinery of the cells to insert in this locus a gene of interest that has been delivered with a non-integrating viral vectors. And for hematopoietic stem cells, we are mainly using adeno-associated viral vectors. I'm going to show you some preliminary data on the genome editing for one of those PIDs called the X-linked agammaglobulinemia, or XLA. This is an antibody deficiency. XLA affects 1 in 200,000 live births and is caused by mutations in a gene called the Bruton tyrosine kinase that is important in B cell development. In fact, B cells start their development in the bone marrow, and at the stage of a pre-B cell, they express what is called precursor B cell receptor, which signals through BTK to progression to maturation into immature B cells. And patients have absent BTK or a non-functional BTK, and this leads to the stop in the development of the cells at the pre-B cell stage. And so patients have absent B cells in the blood and no detectable immunoglobulins, and this leads to severe and life-threatening infections. So the mainstay of infection prevention is immunoglobulin replacement therapy, which mainly stabilize the patients. But this treatment is quite expensive, and some patients suffer complications such as sinopulmonary infections, chronic inflammatory bowel disease, a high incidence of bronchiectasis, and so experience long and frequent hospitalization times that really impact on their quality of life. So scientists are now developing gene therapy for XLA. But one thing we need to remember when thinking about doing a lentiviral, for example, gene addition strategy for this disease is that too little BTK expression may not restore B lymphopoiesis, and on the other hand, too much BTK expression carries the risk of oncogenesis, so it can lead to cancer. And BTK expression is highly regulated during B cell development. So how to achieve this physiological regulation? By gene editing. So our strategy is to use a Cas9 ribonucleoprotein complex to create a double-strand break in the early exon of BTK in exon 2, and then to use AAV6 to deliver the donor template, which is mainly the whole codon-optimized BTK gene from exon 2 onwards, containing also a strong Cossack sequence and a piece of intron 18, which has been shown in other papers for other genes. The last intron is quite important for recognition of the polyadenylation site, so for termination of RNA. So to check if we could restore physiological levels of expression, we used a B cell line called the DG75. This is a Burkitt lymphoma cell line. And we knocked out BTK in this cell line using the CRISPR-Cas system, but this time targeting exon 3. And then we derived clones. And as you can see by the IMAGE analysis, which is reported in the histogram in the bottom, all the clones have a very similar level of BTK expression if compared to wild type, for example. So more importantly, Giorgia T cells, which do not normally express BTK as primary T cells, continue not to express BTK even if they have inserted our integrated transgene, and underneath there is an in and out PCR to check for integration. We then move on to try our protocol in those hematopoietic stem cells, which are called CD34 cells from donors. And so the protocol sees an activation of cells for 2 days, and then the electroporation of a high-fidelity Cas9 complex with a chemically modified guide RNA, and then 15 minutes later, the transduction with AAV6. And this time we used a reporter cassette gene, which is a green fluorescence protein, to readily identify the cells with the integrated event by just looking at the GFP percentage. Also, the levels of CAT, so the levels of indels were studied by tide analysis, and the viability has been reported. So these are data from 3 different donors. And as you can see, the indels percentage is pretty high for all the donors. That means that the CAT is efficient. Viability is also between 60 to 80% between the donors. And the GFP percentage is around 40%. And we need to remember that in XLA, there is a selective advantage. So this level of correction will be beneficial. We then tried our gene editing protocol, again, in healthy donors with the BTK cassette, which is a bigger cassette. And we see with this cassette a bit less viability, especially if looking at day 5, and we now it's important to actually look at viability at a later time point rather than day 2, because day 2 could be misleading. And we see quite a variable HDR, but on and all the percentage of integration ranges between 30 to 60% in some donors. We showed our results to XLA patients that, who were quite excited about our research and decided to donate their cells. And we optimized in vitro a B-cell differentiation protocol, which is a feeder-free culture protocol, where we put our CD34 cells in the presence of IL-6 for 1 week. And then we put the cells on ICAM-coated plates in the presence of IL-7 for another week. And by week 2, we should start having pre-B cells in the culture that are independent on IL-7. So we can remove cytokines and then let the cells grow for further 2-3 weeks, where we can find actually now immature B cells as defined as CD10 positive, CD19 positive, and IgM. And just to remind you, this is the developmental block seen in XLA. So we have performed our gene editing protocol in 1 XLA patient, and I'm going to show you now the in vitro B cell differentiation with those cells. And if you focus on the CD19 positive and CD10 positive gate, you will see that this gate gets populated more and more during the weeks for an healthy donor sample. And then at week 4, this gate is contains a lot of pre-B cells that can also express IgM. So those are properly immature B cells. And while if you look at the plots underneath, so if you look at the XLA patients, there are no immature B cells. And now you can appreciate that gene editing is able to rescue the B cell differentiation defect in these patients, as the plots, the FACS plots, are much more similar to an healthy donor situation. Only 1 month ago, we could put some error bars. We had the second patient donating the cells, and the viability between the patients, in the 2 patients is pretty similar. The gene editing protocol does not affect the ability to form colonies of HSC cells. And here again is to tell you that in both patients, we can finally see immature B cells. And so that tells us that the gene editing relieves the block in maturation in both patients. So now we have nice tools. And so we have a strong proof of concept. And Alessia is going to show you how to take a proof of concept to clinical application. So I will stop sharing.
Okay, thank you. What I was just saying is that we design our lab is focused on designing gene editing strategy to treat Wiskott-Hodgkin syndrome, which is another primary immunodeficiency. And we do so by using a very similar approach to what Giorgia has already showed you. So we aim to knock in a functional gene, in this case for Wiskott-Hodgkin syndrome is the WAS gene, into its own locus to make sure we have endogenous regulated expression of the gene. So to do so, we have designed guide RNA to exon 1 of WAS and introduced the gene with an AAV6 donor vector. So this has been published already, the proof of concept study last year, and we show high feasibility and efficacy of the system. So what we wanted to do next is to make sure that from proof of concept study, we are able to translate this platform into a clinical setting. This will be helpful also to establish a platform to translate gene editing technology in a much wider range of HCC disorders, like, for example, XLA, as just presented by Giorgia. So to do so, what we plan to is to optimize HSPC gene editing protocols to current GMP compliant standards. Then we aim to scale up the gene editing protocol for clinical readiness in order to mimic what will happen in a clinical setting. We also want to assess the functionality of these cells that are large scale manipulated in vitro. And then, of course, we want to evaluate the toxicity of the procedure in vitro and in vivo. So to do so, first of all, we needed to move all our research-grade reagents into clinical-grade reagents. So we first designed a clinically compliant AAV6 donor vector by introducing a kanamycin resistance gene in the vector backbone in place of the ampicillin one, and also shortening the homology arms to facilitate detection of the knock-in cassette in vivo. We tested these vectors in vitro in HSPCs. We couldn't detect any difference in HDR compared to research-grade reagents. And also, to match the planned clinical manufacturing process, we really need to move to clinical-grade reagents and cell culture and gene editing reagents, especially for the Hi-Fi Cas9 and the guide RNA. And so we use those reagents, and we show that HDR rates are stable even when using clinical-grade reagents. Given the high costs of the reagents required for large-scale manufacturing of HSCs, we tested decreasing doses of RMP and cell concentration from product manufacturing, and we found optimal and constant HDR rates when halving the amount of RMP used in the proof of concept study. And lastly, we wanted to test a GMP-compliant max site flow electroporator system, which allows scalable cell electroporation. And we demonstrated comparable and reproducible rates of targeted integration, up to 65% of the cells, when using the clinical-grade max site electroporator, with minimal toxicity compared to the technologies that we use in small scale in proof of concept study, and also compared to unmanipulated cells. So having established a clinical-grade GMP protocol, we wanted to move forward and scale up the whole platform. So we purchased apheresis products of Plerixafor mobilized peripheral bloods, and we freshly enriched for CD34 positive cells, either manually or with an automated Clinimax Prodigy system. We processed 5 apheresis so far with great results in terms of viability and CD34 positive purity. Then we once purified, these cells are cultured for 2 days, and then at day 2 cells are collected. They undergo gene correction protocol adapted for large-scale manufacturing, and then the final product is harvested at day 4 for quality testing, such as viability, editing efficiency, and genotoxicity analysis. Then a small portion of the product is kept in culture for further 2 days, for the 2 weeks, for time course analysis of gene editing and genotoxicity, while the majority is then cryopreserved and then again defrosted, QC analyzed, and then transplanted into animals to mimic a potential clinical process. So here I will show the results that we obtained so far. So we managed to successfully integrate the WAS gene in roughly 60% of CD34 positive stem cells when editing an average of 115 millions of cells, so in large scale, and up to 75% when editing in medium scale, meaning an average of 5 millions of cells per run. HDR rates were stable throughout the in vitro culture and also in cryopreserved product after defrosting and upon infusion into animals. And also indel rates were very high, up to 95% and stable in vitro. Concerning toxicity, gene editing process showed minimal toxicity at a large scale. We have more than 80% of cell viability before, both before and after cryopreservation of the product, and we see negligible differences compared to unmanipulated controls. Importantly, all groups are able to give rise to colonies in CFU assays without lineage skewing, and most importantly, without any difference in terms of numbers of colonies among the groups, indicating that cells are still able to repopulate. So to sum up this part and demonstrate the feasibility of our manufacturing process, we compare our results with the batch release criteria set by our GMP at UCL for the release of gene therapy products. And we show that we are able to meet and actually exceed all key criteria of a good product. After this, we wanted to test the ability of these manufactured large-scale products to engraft long-term and reconstitute hematopoiesis. So to this aim, edited hematopoietic stem cells are injected intravenously into sublethally irradiated NSG mice. And then we evaluated bone marrow human chimerism, targeted integration in the frequencies of targeting in the hematopoietic compartment at 16 weeks post-transplantation. As shown in this table, we have treated a total of 50 mice transplanted with 250 millions of cells to mimic what would happen in a clinical setting if we were to transplant these cells into WAS, into children affected by PID. So here is the results of the in vivo data of mice transplanted with large-scale manufactured cells. Overall, RMP only and WAS edited cells show robust engraftment capacity with a median human chimerism in the bone marrow around 40% for edited cells. So while the engraftment rate is slightly decreased compared to unmanipulated cells in the bone marrow, we can see comparable, if not higher, engraftment in the periphery. When they're looking at the bone marrow, the human graft was multilineage, consisted mostly of B cells and myeloid cells as usually seen in these mice. And when looking at the stem cell compartment in the bone marrow, we can see comparable chimerism among groups of stem cells and more primitive stem cells. Lastly, we also sorted human engrafted cells in the bone marrow, and we detected 15 to 20% of cells harboring a corrected knocked-in cassette after 12 weeks of transplantation. So last but not least, actually very important, we wanted to, one main aim of the preclinical study is to assess safety of gene editing. We all know that one potential risk of gene editing is introduction of unwanted genetic modification that may in turn lead to the development of malignancy. So we really wanted to take a comprehensive approach for evaluating the safety and the toxicity of the gene editing strategy using a combination of sequencing-based analysis and pathology assays. What we did, so cells were manipulated and then were edited again, and some of them were kept in culture and some of them were transplanted into mice. So concerning the in vitro part, we performed off-target analysis soon after the editing process at day 4, and then again at day 14. And at these time points, we also collected samples for karyotyping to detect gross chromosomal rearrangement and also to determine the impact of gene editing on cell fitness. We also performed global gene expression measurement by RNA-seq. At day 21, we also performed Cas-seq analysis to check for chromosomal rearrangements and also ITR-seq to detect AAV integration genome-wide. The same analyses were performed at the end of the transplantation on sorted human cells, and we also wanted to do a histopathology analysis to detect signs of toxicity in the animals due to the gene editing procedure. So sampling and off-target analysis at different time points, especially after transplantation, allow us to identify not only the presence of unwanted genetic modification in the product, but also to analyze the functional relevance of these off-targeting, such as their effect in positive or negative selections of cells bearing specific off-target modifications, the potential of developed malignancies, or exert a clonal dominance effect. For the off-target analysis, we focus our attention on a group of predetermined off-target sites that we preliminary screen in our proof of concept study in small scale. So we indeed screen 15 predicted off-target sites in WASP patient cells samples, and we selected one site out of 15, which gave a very low frequency of indels in just one HSPC donor source. In parallel, we also perform unbiased genome-wide guide-seq analysis, and we retrieve a total of 6 sites, which have been added to the pool of sites to be screened in the large-scale products. So we perform deep sequencing on genomic DNA extracted from large-scale gene-edited cells at day 4, and we could not detect any off-target cutting and any of the sites analyzed when compared to unmanipulated cells. The same analysis was also performed at day 14 after in vitro culture, and again, we could not detect any unwanted genetic modification, further confirming the safety of the platform and an absence of any growth advantage of indels-bearing cells in vitro. We also perform Cas-seq analysis to check for gross chromosomal rearrangement and translocations, and again, we could not detect any major modification. These results were also backed up by a preliminary karyotype analysis that we performed in these cells. Last, concerning the genotoxicity in vivo, much of this part is currently ongoing, so I can't give you a complete view of the results today, but what we have observed so far is that off-target analysis on human cells extracted from the bone marrow of transplanted mice return no indels at the sites analyzed, further confirming the accuracy of the in vitro analysis, but also the absence of any selective advantage on cells bearing unwanted mutations. From a toxicology point of view, no mice experience weight loss after bone marrow conditioning and transplantation, and overall, all mice gained weight regularly and survived until the end of the experiment. And moreover, hematological analysis showed that the total count of blood cells were similar between groups, suggesting absence of major toxic effects. And hopefully this will be confirmed by the pathology analysis on bone marrow smears, as well as spleen sections that we are performing now. So to conclude, we have shown that large-scale manufacturing of hematopoietic stem cells is feasible, with more than 60% of knock-in of the cassette and more than 80% of viability in our case when targeting WASP gene, maintaining the same efficiency that we actually obtain in a small-scale proof-of-concept study. We also show that manipulated cells retain their ability to repopulate the bone marrow of recipient mice, and that there's no evident toxicity so far, as well as no off-targeting activity. And we are now, since we would like to see the toxicity of the platform in a disease model that recapitulates the Wiskott-Hodgkin syndrome disease, we will repeat efficacy and safety studies in a humanized WASP knockout model, where we are actually replacing the WASP murine locus with a human WASP locus. And this will allow to test our gene editing reagents already already used for human locus into a relevant mouse model of the disease. So last, I want to thank also on behalf of Giorgia, all the people that were involved in my team and in Giorgia's team, especially Asma Nazim, Wisdom Vetaroy, Rajiv Rai, and Samir Bahal, especially for the XLA part. A big thank to the GMP facility in our institute that made possible the scale up and preclinical assessment of our gene editing platform. A big thank to the funders, and a big thank to all of you for listening to us today. Thank you.
Okay, I'm just gonna start. All right, my name is May Daher. I'm an assistant professor at MD Anderson in the Department of Stem Cell Transplant and Cellular Therapy. And I would like to thank the organizers for the invitation. And it's my pleasure to be here today to talk to you about CRISPR-optimized cellular therapies. So these are my disclosures. And I won't spend too much time on the background since the audience is very familiar with CRISPR and what it does, basically. But we all know that CRISPR has revolutionized the field of molecular biology and has won the Chemistry Nobel Prize in 2020. And if we think about it, really, it was developed just 9 years ago, and the first results were published in Science since 2012. And since then, there has been an explosion of use of CRISPR in multiple fields, including biology, chemistry, medicine, agriculture, etc. So the scalability of this approach is amazing, and it's due to the elegance, the simplicity of the approach, and its reproducibility. And it really relies on these two simple components, the Cas9 protein, which acts as a scissors to cut the DNA in a double-stranded fashion, and the guide RNA, which is a small piece of RNA that binds to the Cas9 protein and guides it to a specific site of the DNA to be specifically cut. So from a clinical standpoint, really, there has been also an explosion of clinical trials recently testing ex vivo and in vivo CRISPR gene editing strategies to treat multiple diseases, including refractory cancers, including sickle cell beta thalassemia. You've heard about immunodeficiency syndromes as well, and more recently, even amyloidosis. And if you go to clinicaltrials.gov and search for CRISPR gene editing cell therapies, you can find over 15 clinical trials describing these approaches. Most of them are ex vivo clinical trials. Most of them are phase 1 and testing this approach in CAR T cells or hematopoietic stem cells. So at MD Anderson, we're very interested in CRISPR technology as well to enhance the function of our adoptive cellular therapies, and we are mostly focusing on virus-specific T cells and NK cells and CAR-NK cells, so two different platforms, and I'll give you examples of both. So I'll start with the CRISPR-modified virus-specific T cells. We all know that viral infections remain a leading cause of morbidity and mortality after stem cell transplantation and in patients who are immunosuppressed with cancer. Prophylactic therapies such as letermovir or ganciclovir are effective in reducing somewhat the risk of these viral infections, but however, breakthrough infections still occur. And the available pharmacotherapies such as ganciclovir and cidofovir, unfortunately, are limited by their toxicity profile. For example, ganciclovir can cause marrow suppression and cidofovir can cause renal toxicity. So alternative approaches to treat these viral infections are very important, and virus-specific T cells have already been proven to be safe and effective in treating these post-transplant viral infections, including CMV, BKV, adenovirus. There are multiple clinical trials at our institution and other institutions who have proven the safety of this approach and the efficacy, showing like response rates in the 70 to 80%. With no significant adverse events and no graft versus host disease, which is very important in the clinical setting. So at our institution, we generated these biobanks of virus-specific T cells, starting from patients who have recovered from certain viral infections, let's say who are seropositive for BKV. We would isolate the PBMCs, co-culture them with viral PEP mixes and cytokines to generate these virus-specific T cells, and then freeze and generate these biobanks of VSCs to be used when the patients need them. And we have published our experience with BKV-specific T cells treating JC virus-related progressive multifocal leukoencephalopathy, and more recently also published our experience with BKV-CTLs treating BKV cystitis, again, in both settings. Both for PML and BKV cystitis, these BKV CTLs have proven to be very safe and very effective with response rates in the 80% range. So during the pandemic, we started thinking also, why don't we apply this technology to treat COVID-19? So following the same approach, we developed a methodology to expand virus-specific T cells from recovered donors. So we start with a patient who has recovered from COVID-19 infection, isolate the buffy coats, cycle the peripheral blood mononuclear cells, and then co-culture them with viral PEP mixes that span the spike protein, membrane protein, and nucleocapsid protein of the COVID-19 virus. And then also with cytokine cocktails such as IL-2, 4, and 7, and then generate these virus-specific T cells that are specific killers of COVID-19 infected cells, expand them further, and then freeze and generate these biobanks again to treat patients with COVID-19. So after proving the success of this approach in the preclinical setting, we initiated a clinical trial at MD Anderson. It's a phase 1 clinical trial testing this approach in patients with mild COVID-19. So far, 15 patients were treated to date. We have proven the safety of the approach, and we have seen efficacy. All the patients who have been treated on this trial have been discharged from the hospital in good condition, and we have not seen any evidence of cytokine release syndrome or graft versus host disease or any other adverse events. However, despite the success of virus-specific T cells, there remains a very common clinical problem that we encounter in the clinic, and that's the use of corticosteroids. Corticosteroids, as we know, are very commonly used in the post-transplant setting to treat multiple complications such as graft versus host disease. And unfortunately, the use of corticosteroids can cause further immunosuppression and lead to increased risk of viral infections. Steroids also, as we know, are being used for the treatment of COVID-19 infection, especially in moderate to severe disease, in order to prevent lung inflammation and scarring. So the problem is, while these patients are being treated on steroids, we cannot enroll them on the clinical trials testing the safety and efficacy of virus-specific T cells. Why? It's because steroids are usually lymphocytotoxic, therefore they can cause apoptosis and killing of virus-specific T cells. So usually these patients are immediately excluded from the clinical trials if they are on steroid therapy. So a possible solution we thought of is to use CRISPR technology to delete the gene that encodes for the glucocorticoid receptor in virus-specific T cells. So the gene that encodes for the glucocorticoid receptor is called NR3C1, and we wanted to use CRISPR to knock it out. And we asked ourselves 2 questions. By doing this, do we really render these virus-specific T cells resistant to steroids? And do we affect their function or phenotype by doing so? So to answer the first question, we designed 2 different guide RNAs to target NR3C1. And we obtained a very efficient knockout of the glucocorticoid receptor, as you can see at the DNA level by PCR in the A panel, and on the protein level by Western blot in the B panel. You can see a complete loss of the protein. Functionally speaking, these GR knockout virus-specific T cells are completely resistant to steroid, as you can see in the C panel. If you focus on the upper panel here, you can see that the control virus-specific T cells, after treatment with dexamethasone, most of them become either apoptotic or dead, as you can see by annexin 5 staining and live death staining. So the steroids are killing the virus-specific T cells. But when we knock out the NR3C1 gene to make these virus-specific T cells resistant to steroids, indeed, after treatment with dexamethasone, you can see that most of them, all of them, retain their very good viability and do not undergo apoptosis. So this was very encouraging. This is further quantified in the D panel. You can see the percentage of live cells and the control virus-specific T cells after addition of dexamethasone, dramatic drop in viability. However, the GR knockout virus-specific T cells in green and purple after the addition of steroids, you can see that they maintain their viability very nicely. Do we affect their phenotype or their function by doing the GR knockout? We don't. After GR knockout, we maintain the phenotype of these virus-specific T cells very nicely. The distribution of CD4 to CD8 is very similar after the knockout, and the expression of markers such as CD62L and CD45RA is also very similar following the knockout. The ability of these cells to produce cytokines such as interferon gamma, TNF-alpha, and IL-2 is also not affected after the knockout. Even after addition of the steroids, you can see that they maintain their ability to produce these cytokines after exposure to the peptides. So we really were encouraged by these preclinical results, and we wanted to scale up this approach to take it to the clinic. So we used a 4D nucleofactor, Lonza nucleofactor, and we scaled up this approach, starting from 3 million cells, going up to 25 million and up to 100 million cells. And we saw that the knockout efficiency remains very good, even at the 100 million dose level. You can see up to more than 95% protein loss, which was, again, very encouraging. And the second important thing you need to look at for a clinical product is its ability to expand ex vivo. And we were initially, wanted to know if by knocking out the the glucocorticoid receptor, do we affect the ability of these cells to expand? And luckily we don't. So you can see if you compare the solid lines to the dotted lines being the GR knockout virus-specific T cells, and the solid lines representing the wild type or Cas9 control virus-specific T cells, you can see that their ability to expand is very similar at the 3 dose level, 3 million in green, 25 million in blue, and 100 million in red. So again, this tells us that this approach can be scaled up in our GMP facility and can be taken to the clinic. In order to manufacture a clinical product, you need GMP grade materials. So we would use the high fidelity Cas9 called SpyFi, which is GMP grade provided by Aldevron, and a GMP grade guide RNA from Syntigo to target the glucocorticoid receptor gene. So using these materials and using the scale up in our GMP facility, we're already preparing for this next generation clinical trial. Testing GR knockout SARS-CoV-2 specific T cells. So again, following the same schema, starting from a recovered donor who has recovered from COVID-19 infection, getting the buffy coats, the peripheral blood mononuclear cells, co-culturing them with the viral pep mixes and the cytokine cocktail to generate virus-specific T cells against COVID-19, and then introducing this large-scale knockout of the glucocorticoid receptor, and then expanding these cells further before we can freeze and biobank them in our GMP facility to be able to produce these off-the-shelf virus-specific T cell products that are resistant to steroids. And we are planning to use these cells in patients with moderate to severe COVID-19 infection, these are the patients usually who are treated with steroids to prevent lung inflammation and scarring. So we're very excited about that. The trial has been IRB approved and is being reviewed by the FDA. And another clinical trial also testing GR knockout virus-specific T cells to treat stem cell transplant-related infections such as CMV, BKV, and adenovirus is also underway. So moving on to the second part of this talk, focusing on CRISPR-modified CAR-NK cells. As you know, at MD Anderson, we have a platform to engineer natural killer cells or NK cells from umbilical cord blood. So we obtain umbilical cord bloods, we isolate the cord blood derived NK cells from them, and then we expand them with cytokines and feeder cells, and then transduce them with a retroviral vector to express a chimeric antigen receptor on their surface, and also produce cytokines such as IL-15 to enhance their persistence and proliferation. This was the first in human clinical trial led by Dr. Rizvani at MD Anderson. And you can see that in this particular clinical trial, this was the CD19 CAR product. We expand the cells after the CAR transduction, and then the patients in the meantime would be receiving lymphodepleting chemotherapy with cyclophosphamide and fludarabine. And then on day 0 would receive the cell infusion. This was a very successful phase 1 clinical trial where we saw 8 out of the 11 first patients enrolled on the phase 1 arm achieve a complete remission. These were very heavily pretreated patients and with a difficult diagnosis such as Richter's transformation, who have failed multiple previous lines. And in multiple patients, we saw complete remissions. This is the example of patient number 5, who prior to admission, you can see the PET scan lighting up like a Christmas tree. And at 30 days post-CAR and K-cell infusion, you can see complete resolution of these PET-avid lesions. So despite the success of this program, we are starting to see relapses after CAR and K-cell infusions, very similar to what we see in the CAR T-cell field. So these cells are effective, but eventually some of the patients relapse. So we want to see if we can enhance the function of our CAR and K-cells even further by targeting checkpoint molecules. And the first checkpoint molecule we went after was cytokine-inducible SH2 containing protein, or CIS. This protein is encoded by the CIS gene, and it's a negative regulator of JAK-STAT signaling in both T cells and NK cells. And it's been shown very elegantly by Nick Huntington group in Australia that knocking out CIS in murine NK cells can release the brake on their activity against tumor cells. So we wanted to do the same, but with human primary NK cells that have been engineered with chimeric antigen receptor. And we were able to show that this approach is very effective in vitro, but also in vivo in a Raji lymphoma mouse model. We saw that these CIS knockout CAR NK cells can improve tumor control and survival in the mice. And this is a non-curative mouse model, so usually the mice really don't survive beyond a couple of months. But some of the mice that received the CIS knockout CAR infusion were able to survive more than a year. And when we sacrificed them later, we saw no evidence of lymphoma in their organs, as opposed to mice that received the wild-type CAR NK cells or mice that received the Raji lymphoma alone.
Dr. Daler, I think we're a bit over in your session and in the day, so we might want to speed this a little, please.
Yeah, I'm almost done. So taking this approach hopefully to the clinic, we're preparing a clinical trial testing these next generation CAR NK cells for cancer immunotherapy, starting with cord blood, isolating the NK cells, CAR transducing them, and then expanding them, and then introducing this large-scale CRISPR knockout to delete some of the negative regulators of NK cells, such as CIS, TGF-beta receptor 2, and other negative regulators that we're going after, expanding these gene-modified CAR NK cells and then freezing them for use, for off-the-shelf use when the patient need them. So in conclusion, CRISPR-Cas9 technology is a very powerful technology that can be applied to enhance the function and activity of adoptive cellular therapies. And we have proven the efficacy of this approach in both virus-specific T cells and CAR NK cells. And we're very excited for the next generation clinical trials that are in preparation. So with that, I would like to conclude and thank everyone who has been involved in this work, as delineated on this slide, and then especially the patients who have been involved in the clinical trial. Thank you very much for your attention, and I'd be happy to take questions if there's time. If not, then I can answer the chat. Thank you.
Yeah, thank you for that wonderful presentation. There are many questions, so we'll have to jump to the archive and the Q&A. And so, yeah, please look for your answers there. Thank you so much. With that, I'll jump to the next speaker. Dr. Caroline Kuo is the Assistant Professor of Pediatrics in the Division of Allergy, Immunology, and Rheumatology at UCLA. Her research investigates novel therapies for rare immunologic diseases. And with that, please jump in.
So thank you very much for the opportunity to speak today. So I'll be speaking on site-specific gene editing of human hematopoietic stem cells for the treatment of X-linked Hyper-Igm syndrome. This syndrome is characterized by the inability to switch from production of antibodies of the IgM to IgG, IgA, or IgE serotypes. And the reason for this is because in order for a B cell to produce class-switched immunoglobulin, it needs to communicate with a T cell through various co-receptors. But an absolute requirement is between CD40, which is constitutively expressed on B cells, and CD40 ligand, which is expressed on T cells upon immune activation. And so defects in CD40 ligand are what account for the X-linked form of disease. And because CD40 ligand is expressed also on monocytes, macrophages, and dendritic cells, patients present not just with a antibody deficiency, but with a combined immunodeficiency, manifesting as recurrent bacterial sinopulmonary infections. 40% of patients present with pneumocystis jirovecii pneumonias, invasive gastrointestinal infections, oftentimes with cryptosporidial species, autoimmunity, and cancers. And so on the left is a survival probability curve for patients with XHIM. And you can see that the long-term outcomes for these patients remains relatively poor, with about 30% survival at 30 years of age, despite modern medicine. And we know that allogeneic bone marrow transplant is curative. However, because patients generally do well on antibody infusions towards the beginning of their life, this is often postponed until later. And at that time, then the question is, there's unstandardized transplant timing and conditioning regimens, but also patients can now have reactivation of chronic infections, worsening of pre-existing lung damage, and these patients often tend to suffer from graft versus host disease when they undergo allogeneic bone marrow transplant. And so this is a diagram of the CD40 ligand gene showing known mutations that cause the disease. And since all known mutations are located on a single gene, this makes this defect an ideal candidate for gene therapy. There have been two groups who have previously looked at the role of gene therapy for XHIM using mirroring models of the disease. In both, they were able to successfully achieve immune reconstitution by delivering a normal copy of the mirroring CD40 ligand cDNA using retroviral vectors. However, 2/3 of mice in the top study and 1/3 of mice in the bottom study developed abnormal T-cell lymphoproliferation. And so that really emphasized to us that the CD40 ligand gene needs to remain tightly regulated under control of its endogenous promoter. And so to this end, we decided to use site-specific gene integration as a form of gene therapy for this disease. We started off designing site-specific nucleases. Initially, we were working just with TALENs, but really have fully moved over to the CRISPR-Cas9 platform. So these nucleases would bind to the 5' region of the CD40 ligand gene. This would result in double-stranded breaks that can be corrected either through non-homologous end joining, which is error-prone, or if you utilize the sequences surrounding the cut site as homology arms to flank a normal copy of the human CD40 ligand cDNA, you could promote homology-directed repair and allow integration of this corrective cassette at its endogenous location under control of regulatory and promoter elements. And so we started off by screening several guides that bind to the 5' region of the CD40 ligand gene. We took the top 4 guides with the highest levels of allelic disruption and analyzed them for off-target activity using GuideSeq. And as you can see in the data below, that we really only had one guide that had no off-target activity in K562 cell lines. And so because we had always been interested in moving this towards the clinic, we did delve into the off-target activity of this particular guide a little bit more. And so this included repeating GuideSeq using chemically modified guides from Synthago, as well as both wild-type and high-fidelity Cas9s in both K562 cell lines and KG1As. And as you can see on the top, that there were never any off-target sites identified in any of these samples compared to a known low-performing VEGF guide shown below. We also looked at off-target activity of this guide in primary hematopoietic stem and progenitor cells using Discover-seq. We surprisingly identified 15 putative off-target sites, and even though we felt that these were very unlikely given the high rates of mismatch between these off-targets and our target site, we nevertheless performed RAMP-seq to confirm these off-target sites. And as you can see in the graph below, despite high levels of on-target activity shown in that bar on the very far left, that most of the off-target sites that were identified by Discover-Seq are below the level of detection, the limit of detection for this assay, and those that are within the limit of detection, there is no significant difference between mock-treated and CRISPR-RMP treated samples. And so we moved forward with this particular guide that cuts at the very beginning of exon 1. And we utilized T cells derived from patients with X-linked Hyper-Igm. And so with CD40 ligand shown on the x-axis, you can see that healthy individuals express high levels of CD40 ligand when their T cells are stimulated with PMA and inomycin. Untreated T cells from XHIM patients do not express CD40 ligand, while T cells from patients that are treated with the CRISPR-RMP and an AAV6 to deliver a corrective CD40 ligand cDNA donor now have restored CD40 ligand expression. We also wanted to assess the behavior of this restored CD40 ligand protein to ensure that it behaved physiologically. And in order to do this, we cultured edited cells for a prolonged period of time after editing and let them come to rest and then re-stimulated them at regular intervals. And as you can see in this figure on the bottom left, the light purple dots represent resting cells, while the dark purple dots represent stimulated cells. And this ensured to us that our transgene was behaving physiologically and likely under control of the endogenous CD40 ligand promoter. And finally, we wanted to make sure that our transgene could function properly. And in order to do this, we assayed for its ability to bind to soluble chimeric CD40 in a flow-based assay. And as you can see on the right, these gene-modified patient T cells behave similarly to healthy controls shown on the left. One thing we had always noticed with our transgene was that the MFI of the CD40 ligand expression as well as binding to soluble CD40 was always lower than healthy donor T cells. So you can see that healthy donor T cells are shown here in the green, and our XHIM edited T cells are shown in gray. And so we sought to improve our donor by adding a truncated intron as well as a larger portion of an intron. And you can see that we can restore expression of CD40 ligand and its function to wild-type levels. And this may not be particularly important in an in vitro setting or in a setting in which we can achieve very high rates of gene modification in vivo. However, with current rates of in vivo gene editing, which tend to be on the lower side, this may turn out to be significant. We then moved the system into GCSF mobilized peripheral blood stem cells from healthy donors. And shown here, we can achieve around 25% gene editing in peripheral blood stem cells without significantly affecting viability and fold expansion as measured by trypan blue exclusion 24 hours after treatment. We also evaluated for the ability of these peripheral blood stem cells to form multiple myeloid erythroid colonies by culturing them with methylcellulose media and specific cytokines. And as you can see on the left, with our gene-modified cells shown in the light purple bars, that gene modification does not affect these colony formation compared to mock and AAV only treated cells, and it does not affect clonogenic potential shown on the right. Because we're particularly interested in the ability of peripheral blood stem cells to differentiate into T cells that would ultimately express CD40 ligand, we then evaluated the ability of our gene-modified cells to differentiate in the artificial thymic organoid system. And what the system is essentially a 3D co-culture consisting of our gene-modified stem cells combined with a murine stromal cell line that has been transduced to constitutively expressed notch delta-like ligand, which is essential for T cell differentiation. And so what we're showing on the bottom left is essentially formation of mature CD3 positive TCR alpha beta positive T cells, most of which are double positive, but also result in some mature single positive CD8s, as well as a minority of mature CD4 single positives, and with a mostly naive phenotype shown on the bottom right. When we measure gene integration by ddPCR, you can see that PBSCs that are going into the ATO achieve around 25% gene editing, and T cells that come out of the ATO retain about 15% gene modification. And finally, we move the system into the NSG mouse model. And as you can see in the bottom left, the rates of engraftment as measured by human CD445 of total CD45 is similar in the bone marrow, spleen, and peripheral blood of gene-modified mice compared to mock modified mice shown in the light gray bars. The lineage distribution of cells, so CD3, CD19, CD33, and CD56, are also similar between the two study arms. And importantly, on the right, when we look at gene integration rates in the CD45 positive cells in the bone marrow, we see that about on an average, 8% of cells retain their gene modification. And so as we move this work towards translation, we have been able to fortunately collect peripheral blood stem cells from GCSF mobilized XHIM patient volunteers. And so on the left, I'm showing a similar set of T cell plots as one of my earlier slides, demonstrating that we can achieve good rates of gene integration in this particular patient's T cells, as well as restore their CD40 ligand function by binding to soluble CD40. We were also able to acquire a large number of CD34 cells from this patient with 5 days of GCSF mobilization, shown in these numbers here. And we can achieve relatively high purity of CD34 cells. And similar to healthy donor CD34 cells, we are able to achieve good rates of gene modification without significantly affecting viability and fold expansion. And so to conclude, we believe that gene editing of hematopoietic stem cells is a potential therapeutic approach for X-linked Hyper-Igm syndrome. We know that targeted integration of a normal CD40 ligand cDNA restores CD40 ligand expression in T cells, and as well as its physiologic responses to immune stimuli. And in addition, we're able to gene modify hematopoietic stem and progenitor cells and differentiate them in both in vitro and in vivo models while maintaining gene marking. And so a lot of our current and upcoming work really is probably what a large majority of the field is working on for gene editing of hematopoietic stem cells. And that is to increase homology-mediated gene editing in long-term and engrafting HSC with minimal toxicity. And so, some of this may include, better understanding AAV biology and some of the characteristics that are important for having a high-quality prep of AAV, which in our hands and in the experience of others can be quite different depending on where you have AAV produced. Better understanding of cellular immune responses to DNA double-stranded breaks and AAV and viral introduction of donors. And then perhaps if there are no better ways to overcome some of these immune responses to the exogenous elements, perhaps moving on to a non-viral DNA donor platform to deliver our corrective transgene. And so I want to thank you very much for having me here today. I want to thank my small team shown here in the bottom right. We're a small group, but we are persistent. And of course, I want to thank Don Cohn, who's been my mentor for many years, and I remain very closely working with him. So thank you very much.
Thank you. We appreciate that presentation. And with that, I will jump in. My name is TJ Cradick. I am the Chief Scientific Officer at Excision Biotherapeutics, presenting today on our work developing EBT-101 CRISPR-based therapies for HIV. I need to disclose that this talk might contain some forward-looking statements and that I'm an employee at Excision. And I want to start by thanking the team at Excision, who have been wonderful to work with for the last year and a half. If I don't get to mention that later, we are currently hiring. But I also want to particularly thank our number of different academic collaborators, including those at Temple University, who have a number of publications demonstrating the efficacy of multi-guide targeting of virus, including 2 that I'll highlight today. So I'd like to, in particular, thank Dr. Kamil Kahili and his group at Temple University. At Excision, we're developing CRISPR-based therapies intended to cure viral infectious diseases and improve the lives of chronically ill patients. We've heard a number of talks looking at monogenic diseases, and as we're a bit different, I'll describe some of the unique features of targeting HIV and our approach as it differs from some of these kind of established techniques for targeting monogenic diseases. I'll start off by reminding that CRISPR had a life before gene editing as a bacterial adaptive immune system, including the bacteriophage shown in this cartoon. Doesn't quite do justice to the high number of viruses that are attacking the surface of bacteria. At Excision, we're using CRISPR-Cas again to target virus. This cartoon now shows HIV and the human cells, though in both cases, CRISPR-Cas targets the DNA inside the cells. At Excision, we're seeking to improve the lives of large patient populations with unmet medical need, including hepatitis B virus, herpes, JC virus, and HIV. An estimated 38 million people worldwide are infected with the human immunodeficiency virus type 1. We've been showing this diagram for 17 years, almost part of union rules for gene editing. A single cut marked by these scissors is repaired using the cell's DNA repair machinery, resulting in generally small indels, only a few nucleotide. And this can lead to viral escape. And so I'm excited now to change this up and show the diagram on the right, whereby we're using multiple guides. And in the case for HIV, 2 guides with 3 target sites that can drive these large deletions that have minimal probability of viral escape. And I'll jump to a diagram showing their location. There are 1 guide targets the LTR, and so it has a site in both the 5' and 3' ends, and the second guide targets a well-conserved site in GAG. So cutting at 2 of these 3 sites can result in 3 different inactivations, including the 1 kb, 8 kb, or 9 kb deletions as shown by the boxes in the bottom. I'll jump to a video, which I hope starts to play here, because I like this animation as it depicts the nucleases targeting 2 of the 3 target sites in HIV, such as these targets at the LTRs, with the HIV genome shown in black and inserted into the human chromosome drawn in white. The colorization helps to remind us that the target sites are in HIV, so that no target sites are in uninfected cells, or in cells with HIV excised, as in the first bullet point here on the right. The dual guide viral excision strategy also removes the non-human DNA to return the genome to normal or near normal, and as with a number of the monogenic projects, requires one-time therapy to remove this viral genome permanently. So I'll give a quick intro, and certainly as we're pressed for time, use this illustration of the HIV life cycle and replication cycle to remind that HIV is a retrovirus causing AIDS. And though there are medications fighting transcription and replication, EBT-101 targets the integrated HIV as a one-time therapy. EBT-101 only needs to target the replication competent subfraction of HIV-infected cells. So less than 1/10% of the cells in the body become infected. The majority of the HIV DNA is not intact nor replication competent. So EBT-101 expresses SA-Cas nuclease, dual guide RNAs, and is delivered by AAV9. I want to point to a number of preclinical studies that have been published demonstrating the efficacy of the dual guide approach in human cells, in transgenic and humanized mice, and in non-human primates. One study that I'll discuss is from an NIH-funded work from the Kahili lab and colleagues at Temple and the Gendelman lab at University of Nebraska, demonstrating the elimination of HIV in humanized mice. This study irradiated NSG mice at birth, grafted with CD34 human hematopoietic stem cells, producing human T cells that are broadly susceptible to HIV infection. 4 months after humanization, they were infected with HIV, and then they were analyzed for HIV persistence. They're broken into 4 groups: the HIV controls, the humanized mice treated with laser retroviral gene therapy, humanized mice treated with Cas and guides, and the mice receiving this laser retroviral therapy and CRISPR-Cas and guides. And again, it's a HIV strategy with the dual guides and delivered by AAV, targeting these LTR and GAG sites. The first group of mice that were left untreated, these HIV controls. The second group received a single IV injection of AAV CRISPR-Cas. The third group were administered laser art in blue, and then at the top you can see the group. The CRISPR-only line follows, but this art plus CRISPR treatment in green reconstitute the immune cells with 90% CD4 positive T cells. This graph demonstrates plasma viral load, indicating that after administration of the AAV9 CRISPR-Cas, 2 of the 7 mice showed no evidence of viral rebound at 14 weeks. So this is certainly very exciting that these were functionally cured mice, the first example of this. I'll quickly show this, the panel at the right, where tissue imaging demonstrates removal of the viral genomes that are visible by RNA scope in the controls on the left. And the adoptive transfer of the spleen and bone marrow cells were performed on each group of humanized mice to assess the HIV reservoir levels. And this is certainly a difficult assays to do to determine the HIV reservoir levels. So that's why I'm particularly excited about this functional assay, which demonstrates the functional cure of these HIV infected mice. In the bottom right, the mouse in white were these mice that did not have rebound demonstrated in the previous graph, and bone marrow and spleen cells were adoptively transferred to naive mice that did not show viral infection. And so this was, as I mentioned, the confirmation that there was no latent HIV reservoirs in the tissues of these treated animals, which is certainly very exciting for us. Non-human primate studies were also conducted to assess biodistribution to the sites of the viral stores and to ensure that no significant findings were found related to safety. And the findings, including the higher dose, are currently being written up for publication. Briefly, I'll give an overview that we were able to see ex vivo excision. We saw safety, widespread tissue distribution, and removal of the SIV viral DNA from the monkey genome. So a question that I often get is about the specificity. And so I want to highlight this because it's certainly a bit different. The viral sequences are compiled into a viral database and scanned for target sites. Once we have the target sites, they go back to make sure that they're highly conserved through the viral database. The next bioinformatics step, before we even begin experiments, is also to scan the human genome. There were a few talks today about this type of process, but very few are discarded. The viral guides are quite orthogonal, as I'll mention in a bit, to the human genome. And then, as mentioned for other talks, these go into testing both in tissue culture and in animal models. But what I think is really exciting to see is the viral guides have very similar site in the human genome. So we used a number of different bioinformatic methods, including some of the ones we published in past lives, to look for sites that are similar in the human genome. The total number of differences include the nucleotide mismatch, or the bulges or gaps, the differences in length between the guide and DNA sequence. One aspect of viral targeting that even surprises longtime gene editors is this incredibly low number of sites found. Here in the red box are all the sites that were located with up to 3 differences between the 2 guides in the human genome. And there's only 1. And this one site has 2 mismatches even in the seed. So it's very unlikely that this particular site will be cleaved. So, when we extend this out to 4, 5, or 6 differences, we locate some sites in the genome, but as many less than when we're targeting human genes. And for comparison, I'll show a previously published guide that's targeting beta globin, where we found a great deal more. So this is certainly very exciting. This orthogonal nature of the viral sequences leads to the number of sites output for both guides to be less than one guide targeting many human genes. So we've done a number of different assays for possible off-target effects, including in human cell lines with latent HIV, human primary cells which don't have HIV, so we can see that it doesn't target the human genome, humanized mouse models, and these NHP studies that I described will be published soon. And across all of these, there's no conclusive evidence of any off-target editing or structural variations. So this has been certainly very exciting and certainly matching the bioinformatics that I described before. So I think that the really exciting news is that we're progressing to a first-in-human trial for AVT-101, a phase 1/2a 2-part open-label, multi-center, single ascending dose study. And we can announce the sites and other information, but it'll soon be released and available at clinicaltrials.gov, et cetera. Want to mention, as I said at the beginning, that we're hiring. And so I appreciate your attention and thank everyone. And again, I'll try and look for answers to questions in the Q&A tab that's attached. Thank you.